Hello, everyone, and welcome. My name is Heather Ictanis. I am a principal at Geosyntec Consultants in Columbia, Maryland, and the facilitator of today's webinar on behalf of SERDP and ESTCP. Slide 5. Today's event focuses on DoD-funded research efforts to predict how changing climate conditions impact host-pathogen interactions and vector-borne disease transmission. First, Dr. Corey Richards-Zawacki from the University of Pittsburgh will discuss how predictive models of Chytridiomycosis can be used to evaluate the potential of alternative disease management tactics. Her presentation will be followed by a brief Q&A session. Second, Dr. Brian Allen from the University of Illinois at Champaign-Urbana will present on the predicted consequences of climate change on tick-borne disease risks. His talk will also be followed by a Q&A session. We will conclude the webinar with a longer Q&A session featuring both speakers. The next several slides provide instructions on optimizing your webinar experience. If you have not done so already, download Zoom at the link shown here and provided to you in your webinar registration confirmation email. If you cannot download Zoom, you may view the slides using a compatible internet browser such as Firefox or Edge. By creating a free Zoom account. If you are unable to view the slides, if your screen freezes, try keying Control F5 to perform a hard refresh. If you are accessing the audio through your computer, click the arrow next to the Join Audio button and select Test Speaker and Microphone, and follow the prompts as they appear on your screen. If you continue to experience difficulties, please call into the conference line shown here. You may also submit a comment using the chat box. Please use the chat box only for comments related to technical difficulties. The Q&A option should be reserved for questions for the speakers. In case of continued technical difficulties, you can download a PDF of the slides from the webinar webpage and call into the conference line provided to you in your webinar registration confirmation email. We will also be live streaming the webinar on the SERDP and ESTCP YouTube channel at the link shown here. Today's broadcast will be in listen-only mode. You may submit questions by using the Q&A box on your screen. You do not need to wait until the Q&A period to submit your questions. In fact, we encourage you to submit questions in advance of that session. When submitting your questions, please make sure to add your organization name at the end of your question so that we can identify you during the Q&A questions. With that, I would like to introduce Dr. Kurt Preston, who is the SERDP and ESTCP Program Manager for Resource Conservation and Resiliency. Dr. Preston has tracked his career between civilian, university, and military positions. Prior to his current position with SERDP, he was a faculty member and research administrator at the University of Nebraska-Lincoln, where he led faculty development efforts to improve research competitiveness. In this position, he also worked with technology transfer personnel, academic departments, and colleges to build university research capabilities. In addition, Dr. Preston has served as a member of the Chief of the Army Corps of Engineers Environmental Advisory Board. Kurt, please proceed.
Hi, thank you, Heather. I'm happy to welcome everyone to today's SERDP and ESTCP webinar. SERDP is the Strategic Environmental Research and Development Program established in 1991 by Congress as a partnership between the Department of Defense, the Department of Energy, and the Environmental Protection Agency. SERDP's mission is to identify and address high-priority environmental science and technology opportunities that focus on Department of Defense requirements. SERDP funds both fundamental research as well as advanced technology development that ultimately impacts real-world environmental management. ESTCP is the Environmental Security Technology Certification Program, in which we demonstrate innovative environmental and energy technologies. These investments capitalize on past investments under SERDP or other research programs and are designed to transition technologies out of the laboratory and into the field. Especially important in all ESTCP demonstrations is the ultimate transition to implementation and regulatory acceptance. SERDP and ESTCP are complementary programs, with much of SERDP research occurring at the lab and pilot scale with occasional field efforts, while ESTCP demonstrations are primarily at the pilot and field scale, although occasionally supporting lab efforts are conducted. There are 4 program areas in SERDP and 5 in ESTCP. The Installation, Energy, and Water program area is only in ESTCP, while the other 4, environmental restoration, munitions response, resource conservation resiliency, and weapon systems and platforms, are found in SERDP and ESTC programs and managed jointly by a designated program manager. Our webinar today is focusing on research and demonstrations that were conducted under the Resource Conservation and Resiliency program area, which has essentially 3 main areas of research: natural resources, resiliency, and wildland fire. Our webinar series highlights research and demonstration efforts from each of the 5 program areas. As you can see, upcoming webinars will cover a broad range of topics, including energetic materials and munitions, improved characterization, monitoring, and management of submerged munitions in marine environments, managing AFFF impacts in subsurface environments, and assessment of commercially available fluorine-free foams, and others. The next resource conservation webinar will be offered on August 19th and will feature research efforts on ecosystem model comparison at multiple scales and sites. You can find additional information about upcoming webinars at this link. I would like to also remind you to save the date for the 2021 SERDP and ESTCP Symposium, which will be held November 30 to December 3rd in Washington, D.C. This event will showcase the latest technologies that enhance Department of Defense missions through improved environmental and energy performance. Please consider submitting a poster abstract. Finally, I would like to remind you that a copy of the presentation of today's session can be downloaded from our webinar webpage. We would appreciate it also if you could please take a moment to complete the survey that will pop up on your screen at the end of the webcast. I certainly hope you enjoy today's webinar.
It is now my pleasure to introduce our first speaker, Dr. Corey Richards-Zawacki, who is a professor in the Department of Biological Sciences at the University of Pittsburgh. She is also the director of the Pymatuning Laboratory of Ecology, the university's biological field station. Corey has been studying the ecology and evolution of the interaction between amphibians and their Batrachochytrium, or BD, fungal pathogens for the past 15 years, since an outbreak of Chytridiomycosis in Panama devastated the amphibian populations she was studying as a graduate student. She continues her work in Panama, now attempting to uncover the mechanisms by which amphibian hosts have come to persist and, in some cases, recover from BD infections. Corey completed bachelor's degrees in chemical engineering and biology and a doctoral degree in ecology and evolutionary biology at the University of Michigan in Ann Arbor. Corey, please proceed.
All right, thank you very much. It's my pleasure to speak with you today about our research on the effects of climate change on host and pathogen interactions in Chytridiomycosis. All right, so to give you a little bit of an agenda for my talk today, I'm going to start with some background about this disease system. I'll tell you about our project's objectives and some of our results, specifically results that speak to the ecology of this disease, the impact of climate on host and pathogen physiology, and then projections of disease severity under different climate scenarios. I'll end with some conclusions and implications for management, and then some thoughts on the benefits of our research to the DoD. So one of the big problems that ecosystems face is that as our climate changes, new pathogens are increasingly emerging and spreading. Fungal pathogens are increasingly recognized as drivers of wildlife declines and diseases. Good examples of this include white nose syndrome in bats and amphibian chytridiomycosis, which is the disease system that I'll be talking about today. To effectively steward ecosystems in the face of these changing climates and emerging pathogens, we need to be able to better predict, detect, and respond to disease outbreaks. Doing this will require new tools that can help us to clarify the links between climate, host and pathogen physiology, and disease dynamics. Amphibian chytridiomycosis is a disease that's caused by fungi in the genus Batrachochytrium that invade amphibian skin and cause this disease, chytridiomycosis. There are 2 species of Batrachochytrium fungi that are pathogenic to amphibians. Batrachochytrium dendrobatidis, or BD for short, was discovered over 20 years ago and is now known to affect over 500 amphibian species worldwide. It has caused declines and even extinctions in amphibian species across the globe. The second species, Batrachochytrium salamandrivorans, or B-sal for short, was detected much more recently and right now is affecting salamanders in Europe. The concern for this pathogen is that it may eventually reach the United States, which is a hotspot for salamander diversity worldwide. These fungi colonize the skin of adult amphibians and the mouth parts of larval amphibians. They're transmitted by contact between infected individuals or by contact with contaminated environments. Die-offs in amphibians in the United States have been limited to certain amphibian hosts in certain areas of the US. For example, in the Western US, there have been declines and die-offs in the species that are shown on the figure. As our climate changes, we can expect warming temperatures, but also more variability in temperature going forward. We know some about how climate impacts the host-BD interaction from field studies and experiments that happened prior to the start of our research. So for example, field studies have shown greater risk to amphibians in certain climates, and experiments have shown that temperature influences the susceptibility of amphibian hosts to this disease. We also know that the physiology of the BD pathogen is temperature dependent. So in vitro studies have shown us that high temperatures, so in the range of 28 to 30 degrees Celsius, tend to be lethal to the BD pathogen. And exposure studies in amphibians have shown that the pathogen often can adapt faster to changes in temperature than the host can. One of the big questions for our project is, can the pathogen's physiology adapt to new climates? Amphibians are not defenseless against BD. In fact, they have a suite of defenses that are useful against the BD pathogen. The first line of defense is something we call the mucosome, which is sort of a catch-all term for all the good stuff that's in the mucus that's found on amphibian skin. This includes antimicrobial peptides that amphibians produce in granular glands in their skin and secrete into their mucus. It includes lysozyme and antibodies, and it also includes the microbiome in the skin mucus and the bacterial products that can be active against the BD pathogen. The most vulnerable life stage of amphibians to chytridiomycosis is the newly metamorphosed froglets. These froglets are probably most susceptible to BD because they're still developing many of their immune defenses and many of the defenses that are found in the mucosome. So one of the big questions for our research was, how does climate stress impact the development of the amphibian immune system? The objectives of our project include improving our understanding of ecological factors that impact host and pathogen interactions in this disease system, examining the impact of climate on amphibian immune defenses and the virulence of the BD pathogen, projecting changes in exposure pathways and disease severity under alternative climate scenarios, and investigating the impacts of alternative wildlife management strategies for mitigating disease risk. To study the ecology of Chytridiomycosis, we sampled amphibian communities across field sites that span a range of temperature and precipitation gradients in the US. At each of these sites, we used non-invasive skin swabs to assess infection prevalence and intensity. We also collected skin mucus from leopard frogs, our focal group, for immunological studies. This map shows the locations of our 5 sampling sites in Vermont, Pennsylvania, Tennessee, Louisiana, and New Mexico. The colors in the background of the map show the distributions of species in the leopard frog species complex, which were our focal group for studies of immune function. So to give you some of the results from our field studies, we were able to test over 14,000 animals for BD and B-Sal over 3 years. This includes 30 frog and toad species and 20 salamander species. We detected the BD pathogen at all of our field sites and in almost all of the species that we studied, in all of the species where we had enough samples to really get a good sense for whether they were infected or not. However, we did not detect the B-Sal pathogen at any of our field sites. This is good news because it suggests that the B-Sal pathogen has not yet reached these field sites and is not posing a threat to our salamander species. During the process of our field studies, we were also able to collect 7 new isolates of the BD pathogen, which are now useful for research. And we were able to genotype many more isolates of that pathogen using our non-invasive skin swabs, which I'll tell you more about in a few slides. One of the most striking results of our research is that we found very strong seasonal prevalence of the BD pathogen and also seasonal BD load in our amphibian communities. We found that prevalence, which is the proportion of animals infected, and the intensity of infection peaked in late winter to early spring at almost all of our field sites. The only exception to this was what's labeled as Site A, which is our Vermont field site. And our best guess as to why that site is behaving differently is that amphibians have a very short active period that far north, and perhaps that is causing a different seasonal pattern at that site. Interestingly, in our leopard frogs, where we sampled the mucus, we also found that the defenses vary with season as well. Leopard frog peptide concentrations and also their skin microbiomes varied seasonally, such that amphibians had the greatest peptide concentrations, so the best defenses against BD, just before the peak in BD prevalence and load each year. Also, in Ranitscherichoensis, an endangered leopard frog species from New Mexico, we found that individuals with inferior peptides, so less quantity of peptides secreted into their mucus, were at greater risk of BD infection, suggesting that these peptides are very important in the defense against BD. Prior to the start of our research, it was known that there were 2 sublineages of what's called the global pandemic lineage of BD found within the United States. The global pandemic lineage is the part of the BD lineage that has been associated with declines and extinctions worldwide. So if you can see in the figure on the top, the red and the purple show the distribution of those 2 sublineages of the BD global pandemic lineage prior to the start of our work. One of the interesting things that we found in our study, because we could now genotype the BD pathogen from non-invasive skin swabs, is that there is a great diversity of genotypes in both of these sublineages of the GPL at field sites in North America. So at most of our field sites, we found both BD-GPL1 and BD-GPL2 genotypes, we also found genotypes that look like a mix between these at many of our field sites. So we found a surprising diversity in BD genotypes. All of the genotypes we detected were from that global pandemic lineage. And we also found that multiple genotypes infect amphibian hosts from the same wetlands. One of the most striking comparisons that we made is when we compared the diversity of genetic lineages that we found in this SERDP project to the diversity of global lineages of BD that have been studied thus far. We found an equal diversity just among our SERDP sites to what's been sampled worldwide, suggesting that the pathogen is very diverse here within the United States. To answer the question of whether BD can adapt to new climates, we did two things. We first grew our isolates that we collected from our field sites under different conditions to assess their thermal tolerance and their temperature of peak reproduction. We found differences in these traits among the isolates that we collected from our field sites. We then did an artificial evolution experiment where we grew each of these isolates under current and predicted future amphibian body temperatures and compared their growth and reproduction among differently selected lineages. Lineages that we evolved under future climate scenarios outperformed the current climate lineages more often than vice versa in terms of time to reproduction, infectious zoospore density, and also pathogen population growth. So these results suggest that climate change is not likely to reduce the BD risk for amphibians because the pathogen can adapt to new climate scenarios. We also studied how climate impacts host defenses. To do this, we used experiments to ask how perturbations to the larval developmental environment affect the development of immune defenses against BD in newly metamorphosed frogs. To do this, we used either mesocosm experiments outdoors, where we put tadpoles in these cattle tanks or middle-sized ecosystems, or for one of these, we used environmental chambers in the laboratory to simulate climate warming. The three factors that we investigated were how shortened pond duration, as might happen with changes in precipitation, affect the development of immune defenses. We also looked at how developing under a different non-local climate affect development of immune defenses. And we also looked at how developing under a warmer climate scenario affected the development of the immune defenses in our leopard frog species. So what we found when we raised amphibian larvae under conditions with shortened pond duration is that amphibians that developed as larvae in a drying pond showed reduced survival after metamorphosis, so after they had already become froglets. They also showed lower anti-BD function of their mucosal microbiome, and they developed heavier BD infections when we exposed them to infection as froglets. So there were carryover effects of developing in a drying pond on later life stages of amphibians. In our experiment where we had tadpoles developing under non-local climates, which was a reciprocal transplant experiment, we saw that tadpoles developing outside of their local climate had slower growth and reduced survival to metamorphosis. They also showed deformities, like the one shown here with a kinked tail and some deformities to the hind limbs that were developing. And after they metamorphosed, and when we exposed them to BD, these animals that developed in a non-local climate developed heavier BD infections. And finally, when we raised tadpoles under simulated climate warming, we saw reduced survival to metamorphosis, reduced cellular immune responses in the newly metamorphosed froglets. The new froglets also produced fewer of the antimicrobial peptides that we know are important to the defense against BD, and they also showed reduced survival when we exposed them to BD as froglets. The last part of our research was modeling, and to do this, we used integral projection models, which took into account our field data information on pathogen prevalence and load. They took into account environmental variation and climate variation from our field sites. And they also took into account experimental data that allowed us to assign functions to the relationship between climate variables and BD growth. From these models, we're able to make projections about the prevalence and load of BD and the dynamics of this host and pathogen interaction across our field sites to help to inform management actions. So using our model, we were able to ask how temperature and host demography contribute to BD dynamics. We tested several different hypotheses using our model to see which was a best fit to our field collected data. And the hypothesis that we found to be the best fit was that the dynamics of BD is due to a combination of seasonal breeding migrations of the amphibian hosts, temperature-dependent survival of BD zoospores, and temperature-dependent BD growth. So using this best model, we were then able to ask which amphibian hosts can act as reservoirs for BD, keeping the pathogen in the aquatic environment and potentially making threatened or endangered species susceptible to disease outbreaks. So when we asked how the ability of a host species to act as a reservoir changed, we did this across a variety of scenarios with host population densities and also across each of our field sites. And we found that both host population density and field site were important predictors of whether or not a species can be a reservoir host for BD. We also found that our leopard frogs, our focal taxon, contribute more to BD persistence in aquatic environments during the cooler months of the year. This seems to be because this is the time of year when amphibians are breeding and also when they have high BD loads. So in summary, we find that the ability of species to act as a reservoir host depends on host density and season. Using this information, we then moved on to asking what might be the most effective strategy to reduce BD persistence. The most conventional strategy to reduce persistence of a pathogen is to reduce the seasonal R0 or reproductive value of the pathogen below a value of 1. For our system, this would mean reducing high infection loads during the cooler summer month. And this is a challenging thing to do because it would mean capturing amphibians and applying some sort of treatment to reduce these infection loads, something like a antifungal treatment or perhaps a probiotic. Given the complexity of that, our modeling efforts also suggested an alternative strategy, which would be to reduce the minimum seasonal prevalence of BD. If we can do this, we may be able to cause the stochastic extinction of BD in wetlands, and that would be easier to do than reducing R0 below 1. So some management actions that might be able to help us reduce the minimum seasonal BD prevalence include increasing habitat connectivity, which can help increase dispersal of infected individuals away from the pond environment, and that would reduce transmission of the pathogen. Also, we could increase the availability of thermal refugia for amphibians, because if warm refugia are available, this can help animals increase the rate of BD clearance when they're not in the water, and that would also help to reduce transmission. So some conclusions from our work is that the BD pathogen is ubiquitous and genetically diverse in US amphibian communities. However, the B-Sow pathogen, which threatens our salamander species should it arrive here, was not detected. BD dynamics and the host immune defenses both vary with temperature and across seasons. And we also found in our experiments that climate warming may exacerbate disease risk for amphibians because BD can adapt to new climates, and climate stress negatively affects host survival and defenses, especially of the most vulnerable new froglet life stage. We also conclude that management actions might focus on identifying reservoir hosts and reducing BD persistence in aquatic habitats. Some of the benefits of our work to the DoD include a clearer understanding of the geographic, genetic, and host distribution of BD and B cell, understanding the drivers of seasonal disease dynamics in the system, understanding climate change impacts on disease-relevant host and pathogen physiology. We also are producing benefits by developing empirically derived models to guide management of amphibian communities, including answers to the questions about which species are potential reservoir hosts for this pathogen, and when, where, and how we should manage in order to reduce BD persistence and disease risk for at-risk species. Before I conclude, I want to be sure to acknowledge the large team of collaborators that have generated the data and the insights that I presented today. This includes my collaborators at the University of Pittsburgh, collaborators from the University of California at Santa Barbara, Vanderbilt University Medical Center, University of California, Berkeley, University of Nevada, Reno, and the University of Massachusetts, Boston. For additional information about our project, feel free to visit the website listed here. Also, there is contact information for myself listed below. Thank you very much.
Thank you so much, Corey. And as a reminder, you may submit your questions using the Q&A box on your screen. And we have received several questions on the first part of the webinar that Corey will answer. Corey, so the first question received is, how do you interpret the large amount of genetic diversity you found in the BD pathogen across your field sites?
Yeah, that's a great question. That was something that we were surprised by. I guess not necessarily the amount of diversity across the field sites, but the amount of diversity we saw within a field site was surprising. This much diversity suggests to me that the pathogen has been around in the United States for a while and has been moving around the United States for a while. So we're not seeing a pattern of something called isolation by distance, which would suggest that it's sort of evolving in isolated populations. Rather, it looks like there's a lot of movement potentially of BD lineages from place to place, which is generating this sort of mixing of genotypes across the eastern US. This is in contrast to what we've seen in other parts of the world. So for example, in Panama, where I do research, we think that the pathogen arrived much more recently. And we also don't see this level of genetic variation among sites in Panama. So it's an interesting contrast that suggests that this disease has been around for a while in the United States and is moving pretty rapidly from place to place.
Thank you. The next question, why did your research focus so heavily on the microbiome and peptides in the skin mucus?
Yeah, that's a great question. So Chytridiomycosis is an interesting disease because it really is a skin disease. And so the most important defenses that amphibians have are the defenses that are on the skin. So hence the skin microbiome, which we know from research mainly done by some of my colleagues on our team, that is a home to a diverse array of bacteria and even fungi that produce anti-BD products that can really be helpful in controlling or even getting rid of BD infections. We know from previous work that there is some predictability in terms of what the microbiome looks like, in terms of whether species are susceptible or less susceptible to the pathogen. And also there's been some limited success with manipulating the skin microbiome through probiotic treatments to affect the susceptibility of individual amphibians as well. So we know the skin microbiome is an important line of defense. The antimicrobial peptides are also an important line of defense that's produced by the amphibians themselves. We know that these antimicrobial peptides are diverse, and there have been many studies showing that the suite of peptides that amphibians produce can be a good predictor of how susceptible or resistant they are to infection with BD. So those were the initial factors that we focused on because we had the best evidence going into this project that they were going to be important to understanding the susceptibility of amphibians to BD. We didn't know, however, how they vary across climates or with season, which was a big part of our research.
Thank you. The next question is, what can the results of your project tell us about the effects of climate change on other pathogen outbreaks?
Yeah, that's a great question. I think some of the most relevant systems in terms of wildlife pathogens that our work may speak to are things like white nose syndrome in bats and maybe even colony collapse disorder in bees, where the pathogen's ability to cause disease also seems to be very temperature dependent. This is a common phenomena, especially with ectothermic animals, animals that don't regulate their internal body temperature, which you would think wouldn't include bats, but in fact, when bats are hibernating, they act more like ectotherms, which is when they're susceptible to this white nose pathogen. So I think many of the things that we're learning in this study about the role of, say, the microbiome and also about variability in immune defenses over seasons and how stressors during certain life stages impact the development of immune defenses are things that could be carried over to try to understand how climate may impact some of these other disease systems as well. Also, the modeling approach that we developed that really integrates field studies and experimental data is one that I think will have applications to other disease systems as well.
Thank you. One final question that we've received is, how might the DoD use your findings to help mitigate the impact of BD on threatened and endangered amphibians?
Yeah, that's a great question. So unfortunately, it looks like BD is pretty much everywhere. It's very diverse. We also found differences in the pathogenicity of the different lineages of BD, which suggests that it's going to be hard to predict when you're going to get a very pathogenic lineage. So our modeling results suggest that some of the best things that we can probably do is to try to mitigate the impacts of some of the host species that are sort of more tolerant of the pathogen, can develop high pathogen loads and hence be a reservoir for the pathogen. And so if we can reduce the ability of those reservoir hosts to transmit disease to these threatened or endangered species in the aquatic environment, that seems like one of the best things that we can do. And so some of the things that we had recommended were perhaps improving the connectivity of habitats so that these infected animals have a place to go other than the pond, and they don't sort of ramp up the level of infection in the ponds. Also, potentially doing some sort of treatment or restricting the movement of the more susceptible species so that they don't come in contact with these reservoir hosts could potentially be a good way of moving forward and keeping those more susceptible species protected.
Thank you so much for that. And we'd like to thank you again for your presentation. As a reminder, I'd like to let everyone know that you can use the Q&A box on your screen to submit questions for Corey at the end of the webinar, as well as during Brian's upcoming presentation. And with that, I'd like to introduce today's second speaker, Dr. Brian Allen. He's an associate professor in the Department of Entomology and the School of Integrative Biology at the University of Illinois, Urbana-Champaign. Brian has studied ticks and tick-borne diseases for over 20 years. He's interested in how human-mediated global change, such as climate change and human land use, affect human exposure risk. Since 2016, he's been funded by SERDP to investigate the impacts of climate change, prescribed fire, and plant invasions on risk of exposure to tick-borne diseases on DoD installations in the southeastern United States. He received bachelor's degrees in biology and zoological anthropology from the University of Michigan in Ann Arbor, and a doctoral degree in evolution, ecology, and population biology from Washington University in St. Louis, Missouri. Brian, please proceed.
Okay, thank you for that nice introduction, and thank you to all the attendees today for attending our webinar. I'd like to start by thanking my collaborators on this research, Luke Flory from the University of Florida and Mike Dietz from Boston University, also co-investigator Allison Gardner from the University of Maine, our project manager Waylon Dillon, assistant project manager Drew Hyatt, our climate modeler Tess McCabe, and our molecular lab manager Paige Fredericks, without all of whom the research I'll be sharing with you today would not have been possible. I'll start by giving an overview of the many complex pathways by which climate could potentially impact tick-borne disease risk. And for the region of the US in which we're conducting our fieldwork, the southeastern United States, there are multiple potential pathways by which climate could impact tick-borne disease. And we're particularly interested in fire and plant invasions as these mechanistic pathways by which climate change could have indirect effects on tick-borne disease risk for humans. I'll present to you data from a large-scale survey we performed across 9 Department of Defense installations spanning the southeastern US, where we collected data relevant to this question. I'll also show you results from an experimental study conducted at a site with an extensive invasion of a particular non-native plant species that was a focus of this research, and how both host and tick abundance changed in invaded compared to uninvaded habitats. And towards the end of my talk, I'll share with you our progress towards developing a decision support tool, which is software that can be used by environmental managers to make decisions about management actions that consider some of these interacting effects between multiple factors that land managers may wish to consider in making management decisions. So the pathways by which climate change could influence vector-borne disease risk are diverse. For decades, biologists have been interested in direct effects of climate change on vector-borne diseases being transmitted by ticks, mosquitoes, and other arthropods. And the reason that there's been a suspicion that climate change would impact these disease systems is that the arthropods themselves may be affected by changes in climate, potentially having longer seasons during which to complete their life cycles. Pathogens may also replicate more rapidly under higher temperatures. And so the potential direct pathway of climate change on vector-borne disease risk has received a lot of attention. Increasingly, biologists are aware that there may be other indirect pathways by which climate change may also alter vector-borne disease risk. For example, we know that fire management and wildfire can impact the abundance of ticks and the prevalence of tick-borne disease. Similarly, there are numerous examples now of how invasions by non-native plant species can increase habitat suitability for ticks and increase tick survival. So you can have direct effects of fire and plant invasions on ticks. And then you can also have indirect effects of fire and plant invasions on ticks mediated through changes in wildlife host abundance. Wildlife hosts are critical to the transmission cycles of these diseases, both because they serve as primary hosts for the ticks themselves, but also because they serve as what we refer to as pathogen reservoirs, meaning the ticks become infected with these pathogens as a result of feeding on wildlife. And so how wildlife respond to fire management or plant invasions can also impact tick-borne disease. And so you can imagine then that there's a potential for a cascade of effects through systems as climate changes and alters the distribution of fire in the landscape, the extent of plant invasions, how wildlife hosts respond to those environmental changes, and ultimately then the prevalence of ticks infected with pathogens in the environment that could potentially bite humans and expose them to tick-borne diseases. This is of increasing concern due to the ongoing emergence of tick-borne disease in the US, particularly in the eastern US. We have several ticks that act as vectors of pathogens to humans. Perhaps the best known is the black-legged tick, also known as the deer tick, which not only vectors Lyme disease, but also anaplasmosis and babesiosis to humans. And so there are at least 3 important diseases associated with the black-legged tick that cause considerable incidence in humans throughout the range of this tick species. The Lone Star tick, distributed throughout most of the southeastern United States, is a major vector of ehrlichiosis. The American dog tick, which has a very widespread distribution, transmits Rocky Mountain spotted fever. And an emerging vector in the southeast, the Gulf Coast tick, is also a vector of spotted fever rickettsiosis. Not shown here is an ongoing invasion by a non-native tick species, the Asian longhorn tick, which will unfortunately likely become another major vector of tick-borne disease in the eastern United States. While there are many invasive plants occurring throughout this region that could potentially alter tick-borne disease, for the study I will present to you today, we focused on a widespread and problematic invasive grass in the southeast US, Cogongrass. Cogongrass is a noxious invader of southeast pine forests. And while there are a variety of consequences of these invasions, including biodiversity loss, perhaps the greatest concern associated with Cogongrass for land managers is the way Cogongrass can alter fire cycles. Here you can see images of experimental fires involving native vegetation on the left and Cogongrass on the right. Through creating large amounts of standing dead biomass, Cogongrass can create unusually intense fires that greatly complicate fire management for environmental managers. This is especially problematic on Department of Defense installations where there is munitions, because these can create especially large and difficult to control fires in those environments. And so Cogongrass is a major problematic invasive species from the perspective of Department of Defense installations in the Southeast. I'll start by describing our large-scale survey, which we conducted across 9 installations in the Southeast. We surveyed these installations over 2 years, 2017 and 2018, and established close to 100 plots across these 9 installations on which we sampled a variety of landscape variables. Most of these plots were located in longleaf pine habitats. Our sample plot design is as follows. I won't go into detail on each aspect of it, but to give the overview, we collected information on overstory tree density and diversity, understory vegetation cover and diversity. We also sampled tick abundance using CO2 traps depicted on the lower left here. CO2 trap is a very simple device for sampling particularly Lone Star ticks. It's a lunch cooler attached to a board. You surround it with double-sided carpet tape, place dry ice inside the cooler, and as the dry ice sublimates, it releases gaseous CO2 that attracts ticks, believing that there is a respiring host, and those ticks become ensnared in the carpet tape and are there to then collect from the tape. We also sampled host abundance at each of these plots using wildlife dung surveys, which is a reliable method to assess wildlife activity. I'll show a couple of the major results before giving the overview of how all of these pieces fit together. So from the perspective of items that I think land managers might be particularly interested in, this is the relationship between the number of plots we sampled and years since fire along the x-axis for both invaded and uninvaded. So invaded refers to Cogongrass, and throughout all my slides will be color-coded in red, uninvaded in blue for native vegetation. Interestingly, we see a trend where there are more Cogongrass invasions in more recently burned areas. It's a little hard to tease out cause and effect on this relationship. We know that there's very active management of Cogongrass taking place on DoD installations, but it is a invasive plant species that under some circumstances can become part of a positive feedback loop with fire, where it can become more prevalent as a result of fire. And so this is one of the challenges associated with the management of Cogongrass. The result that I think many will be particularly interested in is the relationship between fire and the abundance of ticks across the 9 installations. Here, not just using time since fire, but a fire return index that takes into account both the frequency and the recency of prescribed fire management along the x-axis, with the most recently and frequently burned sites on the left, and less frequently and less recently burned sites on the right. And I'm sorry, I said the reverse of that, with more recently and more frequently burned sites on the right. And what you can see is that there is lower tick abundance in sites that have been recently and frequently burned. we don't see a significant relation or significant difference between invaded and uninvaded plots, although we had relatively few invaded plots within these DoD sites to sample as a result of aggressive control measures. Putting these results together, we use a statistical approach referred to as path analysis. And what's the main advantage of path analysis is it allows us to tease apart both direct and indirect effects of these interacting variables on our metric of interest, which is the abundance of ticks, a measure of the probability of being exposed to a tick-borne disease. In these path analyses, solid lines refer to statistically significant paths, while dashed lines are non-significant paths, and the numbers you see next to the lines are regression coefficients. What you can see from our path analysis based on our survey of these DoD installations is that fire management has a significant negative effect on tick abundance. And so this is a direct effect of fire reducing the abundance of ticks. Interestingly, though, we also see significant indirect effects mediated via pathways involving leaf litter and canopy cover. Fire significantly reduces the availability of leaf litter. Leaf litter has a positive effect on tick abundance. And so via this indirect pathway, fire reduces tick abundance through the reduction of leaf litter. Similarly, canopy cover has a positive effect on litter, and both canopy and like litter, canopy has a positive effect on tick survival. And so we see indirect effects of fire on ticks mediated through changes in litter and canopy cover, as well as direct negative effects of fire on ticks. As I'll summarize in the next slide, we actually see no significant effect of these variables through wildlife abundance on tick abundance, which is fairly interesting. So to summarize the results of these analyses, we find frequent and recent fire lowers overall environmental tick abundance. The direct effect of fire on tick abundance is stronger than the indirect effect via litter cover, but there is a significant indirect pathway here as well. And in many scenarios, we do typically find a significant effect of host abundance on tick abundance, but in these longleaf pine ecosystems, we did not see a significant relationship with host abundance, suggesting that these abiotic factors are likely the major drivers of tick abundance in Southeast pine forests. We also had the opportunity to conduct widespread sampling of pathogens in the ticks we collected across these 9 Department of Defense installations. This is a sophisticated tool involving next generation sequencing that allows us to screen for a very wide diversity of tick-borne microbes. We're actually able to detect 32 different microbial species from ticks using this tool. Some of these are microbes that cause disease in wildlife, but not in humans. Some are microbes that cause disease in humans, and some are what we refer to as non-pathogenic endosymbionts, meaning they're microbes that are dependent on ticks to complete their life cycles, but are not known to cause disease. We did detect a number of important human pathogens in these samples. We found Rickettsia parkeri in the Gulf Coast ticks that we sampled. This is the main pathogen associated with this tick species. The most common tick in our sampling was the Lone Star tick, Amblyomma americanum, but we also collected Gulf Coast ticks, American dog ticks, a tick I didn't mention previously, the brown dog tick, and just a few black-legged ticks. In addition to finding Rickettsia parkeri in the Gulf Coast ticks, in the Lone Star ticks, we found Ehrlichia pathogens that cause ehrlichiosis in humans. And perhaps the most interesting result from the perspective of these microbes is within the Lone Star ticks, here in dark green, we found a high prevalence of a species of Ehrlichia, Ehrlichia murus oclarensis, which has never been detected in Lone Star ticks before. It was only recently discovered as a cause of human ehrlichiosis in the upper Midwest and associated with the black-legged tick. But this high prevalence that we find for the first time in the Southeast suggests that this could be an important cause of human ehrlichiosis in the Southeast. And we're in the process of publishing that result because we believe it could be important to physicians diagnosing ehrlichiosis in the Southeast. So in addition to this large-scale survey, we also performed an experimental study at a smaller scale to try to tease apart these pathways by which invasive Cogongrass could alter tick-borne disease. There's this sort of direct pathway by which Cogongrass could alter tick survival and thereby alter tick abundance and human risk of exposure to ticks. But there's also indirect pathways potentially mediated through how wildlife hosts respond to these plant invasions. And so to assess these pathways, we collected data on wildlife hosts using dung surveys and wildlife camera traps. We collected data on tick survival inside and outside of Cogongrass invasions using experimental enclosures. And then we also used those same CO2 traps I mentioned in a previous slide to sample tick abundance in Cogongrass invaded and uninvaded plots. Interestingly, we find Cogongrass greatly enhances tick survival. These are assays performed with nymphal and adult stage lone star ticks. Again, the color code is ticks in native habitat are shaded in blue, ticks in the invaded habitat are shaded in red. And we found that ticks placed in Cogongrass plots had 57% higher survival for nymphs and 52% higher survival for adults. And we believe this is mediated by how Cogongrass alters microclimate that essentially alleviates some of the most stressful conditions that ticks experience. If you look at the relationship with relative humidity, 80% is kind of a critical threshold for tick desiccation. Once you get below that value, ticks start to experience water loss, and that leads to desiccation. Ticks, the native vegetation plots basically were below that 80% threshold the majority of the time, whereas the Cogongrass plots spent considerably higher amount of time above that critical 80% threshold. We also see a similar relationship with temperature, where the uninvaded plots experienced higher temperatures relative to the invaded plots. Conversely, we found that hosts generally were more active in the uninvaded areas. And we found an interesting interaction whereby hosts had a significant positive effect on tick abundance in the native plant communities, but not in the uninvaded plots that we sampled. And so this suggests that the effect of hosts on tick abundance is more akin to what we're used to seeing in terms of wildlife tick population relationships in the native plots, but that in the invaded plots, host activity is less, and there's no significant relationship between host abundance and tick abundance in the invaded plots. When we combine these, we see an interesting phenomenon, which is that looking at the data from the traps placed in the invaded and the native vegetations, we don't actually see a statistically significant difference in tick abundance. So there's no significant difference in abundance between the native and the invaded plots. However, we think we understand that this is actually the result of the two different pathways resulting in a similar net outcome. In the case of the invaded plots, it is higher tick survival under Cogongrass invasion that likely leads to the abundance of ticks we find in the invaded sites. Whereas in the native plots where tick survival is lower, it's native vegetation perhaps attracting greater host activity that results in a comparable abundance of ticks in the native plots. And so this is really interesting because it suggests you essentially have offsetting phenomena driving tick abundance in these invaded compared to uninvaded areas. Our work on how climate change will alter these relationships has revealed a couple of interesting factors already. So because we have these 9 installations spanning the southeastern US, we can capture a nice range of temperature and precipitation gradients. This lower plot on the right here shows the range of mean temperatures and the range of mean precipitation for the 9 installations that we sampled across the southeast. And we get a good span that helps us integrate these climate variables into our modeling framework, which is a model called Ecosystem Demography 2, which is a terrestrial biosphere model that can take into account a variety of climate and biophysical factors simultaneously, including hydrology and tree growth. As part of our project into Ecosystem Demography 2, we've integrated Cogongrass as plant functional type, and this has allowed us to start to model how climate change will impact the invasion of Cogongrass. And this is one of our major findings, is that Cogongrass is likely to perform very well under future climate scenarios. And so this is going to be an increasing problem for land managers, that Cogongrass invasions are going to likely become more extensive over time. A second major finding of our climate work is that currently prescribed fire does not appear to be limited by, or strongly limited by, climate conditions at the 9 installations in the southeast. And this is interesting because a number of recent works looking at climate change and prescribed fire suggest that this will be one of the major challenges that land managers chase under future climate, is that the number of days under which prescribed fire can be applied will become less frequent, less available. And so it's going to be more challenging to perform prescribed fire under future climate scenarios. This could lead to a problematic feedback cycle with more wild land fire as a result. We are projecting these effects of climate change on tick-borne disease risk into our novel decision support tool. This is an interactive web interface that land managers will be able to use to make integrated environmental management decisions. So decision support tools are commonly used already by land managers for making decisions about when and where to apply prescribed fire. And in interviews we conducted with land managers at the DoD installations we sampled, we found a number of them are using these decision support tools specifically for fire. What's interesting, however, is that no such tools, to our knowledge, exist either for managing plant invasions or for considering the effects of these environmental management decisions on ticks and tick-borne diseases. And so a major objective of our project then is to develop a decision support tool that integrates environmental management decisions about prescribed fire, plant invasions, and tick-borne disease exposure risk into a single software that will allow land managers to make more comprehensive decisions about how they tackle these challenges. If you're interested in viewing it, we have, here's a link to our draft decision support tool. It's an interactive web tool, and you can zoom in on the installations that we sampled. You can see detailed data concerning tick abundance, pathogen prevalence, or their combination, their product, which is our best metric of tick-borne disease risk. And as we integrate the climate projections, that will allow us to better understand how future climate scenarios will impact tick-borne disease risk. So our research confirms that there are many potential pathways by which climate change can interact with prescribed fire and plant invasions in ways that will alter tick-borne disease risk. The effects of prescribed fire on tick-borne disease risk involve both direct and indirect pathways. In these southeast forests in which we conducted our sampling, we do find that prescribed fire reduces tick abundance. And so this is a potential benefit to land managers of utilizing prescribed fire, is that this in the short term appears to reduce tick-borne disease risk to humans. However, there are complicated relationships with invasive plants, some of which may result in changes that have offsetting effects. As we build these mechanistic models, we can incorporate future effects of climate on tick-borne disease risk, and this will make it possible to incorporate multiple environmental management goals into management decision makings. We see a number of benefits of this research to the Department of Defense. In our interviews with DoD land managers, we asked them what were their highest priority goals. Prescribed fire for habitat management is already a top goal, along with control of plant invasions. As might be expected, it's different invasive plant species at different installations, and how each of these potentially interacts with fire and tick-borne disease risk in many cases is unknown. But at least for Cogongrass, which is one of the most problematic invasive species, we were able to generate a considerable amount of new information on this topic. One thing that was interesting to us is that tick-borne diseases are generally not perceived by land managers as an environmental management goal. They're thought of as an important human health concern. And so one thing we hope we have accomplished through this research is to convey that this is also something that can be managed in the environment. And some of the management tools that land managers are already using may directly impact tick-borne disease risk. And so this is something we hope our project can help convey. We performed this research on Department of Defense installations, and we've been able to relay to the installation managers our findings, including the prevalence of ticks and pathogens on those installations. And we are developing our decision support tool in consultation with DoD land managers to help with future environmental management decision making. For more information about our project, please visit our SERDP website, and my contact information is at the bottom of the slide. Thank you for your attention.
Thank you so much, Brian, for that excellent presentation.
My pleasure.
As a reminder, you may submit questions using the Q&A box on your screen. We received several questions on the second part of the webinar that Brian will answer. Then both Corey and Brian will answer some general questions on how changing climate conditions impact host pathogen interactions and vector-borne disease transmissions. So Brian, as I said, we've received a couple questions for you, and the first one from the Oregon Military Department is, did the analysis account for the quote-unquote clearing effect that deer can have on Lyme disease infections on ticks that feed on deer?
Yeah, I really like this question. So in areas where Lyme disease is prevalent, deer play a dual role. They increase tick abundance as a critical host for ticks, especially adult life stage ticks, but they are not competent reservoirs for the Lyme pathogen, so they don't infect ticks with the Lyme pathogen. We only captured 3 black-legged ticks in the Southeast as part of our surveys. They're considerably less abundant in that part of the country. The main tick we captured, the Lone Star tick, actually feeds on deer in all 3 life stages, and deer are an important pathogen reservoir for Lone Star tick associated pathogens such as Ehrlichia. So in the case of the Southeast, deer are increasing tick-borne disease risk by both pathways. They both increase tick abundance and they increase tick infection rates. The host data I shared was primarily deer, although there were other hosts that we detected, and I showed the data for all hosts combined. But that's one thing that we're still teasing apart, is whether there are some hosts that have, say, a pathogen reducing effect or a tick abundance reducing effect, depending on the key role they play in those transmission cycles. That's a great question.
Thank you. Another question is, should prescribed fire be considered an important tool for managing tick-borne diseases?
Yeah, in my opinion, the short answer is yes. Certainly the relationships we saw from our surveys across these installations in the Southeast was that sites that had burned more recently and more frequently had lower tick abundance. So that suggests that the general pattern for the Southeast pine forests, or at least specifically longleaf pine forests, is that negative relationship. There are examples of tick abundance increasing following prescribed fire as a result of an increase in host abundance. So hosts respond positively to those fires due to an increase in food availability and bring ticks with them. So in some cases, the answer might be a little more complicated, but I think for the installations we sampled, I would generally be comfortable saying that prescribed fire helps reduce tick abundance.
Okay, thank you. Another question is, how will climate change complicate efforts to control tick-borne diseases using prescribed fire?
Yeah, this is, I think, sort of this long-term concern that prescribed fire is going to be more difficult to perform under climate change, and so that there will be fewer days with suitable conditions available for prescribed fire. This could then result in greater buildup of fuels that make those prescribed fires trickier to perform. And so not our work, but work by other researchers suggests that as the climate, particularly as temperatures increase in the springs and falls, when a lot of prescribed fire is performed, gets hotter, those will be more challenging conditions under which to perform prescribed fire, and that could result in more difficult to control wildland fires.
Okay, thank you. What steps should be taken by personnel on DoD installations to avoid tick-borne diseases?
So my understanding is that on a lot of installations under which there's military exercises and training, that personnel utilize permethrin-treated clothing, and that has been shown to be really effective. And so the best strategy is to prevent tick bites in the first place. And so that type of permethrin-treated clothing that in some cases has been developed by the military is really quite effective at preventing tick bites. Also regular tick checks. So after people have been out in areas where they may become exposed to ticks, performing regular checks for ticks is also a good strategy because in many cases ticks have to be attached for a while to transmit a pathogen. This gives some time to remove those ticks before a human can become infected.
Thank you. And then the final question for you, Brian, is how might DoD implement findings from this research to help achieve environmental management goals?
Well, our hope is that we'll be able to provide a tool that will allow land managers to integrate additional management goals into their decision-making on top of prescribed fire. I'm under the impression that these installations in the Southeast, prescribed fire is one of the major environmental management goals. And so a lot of decision-making revolves around that. And we think there's the opportunity to affect multiple positive outcomes through having decision tools that allow managers to take into account the impacts of fire management on, say, invasive plant control and tick-borne disease. And that will allow more efficient use of resources and also hopefully affect multiple beneficial outcomes, including reducing tick-borne disease exposure for personnel on those installations.
Thank you again for answering the questions and for your presentation. And now I'd like to transition to a more general question and answer session for both Corey and Brian to respond to questions that we've received. So for the first question, Corey, could you respond to which aspects of climate change are most important to predict accurately in order to mitigate impacts on emerging infectious diseases? And then after Corey is done with her response, let's turn the mic over and hear from Brian.
Yeah, I'm happy to contribute to that question. It's a tough one, I think, because unfortunately, I think some of the things that are hardest for us to predict are the things that will probably be the most useful for us to know. Like when I was talking about the impacts of climate change on global temperatures, we will see an increase in global, air temperature, but we're also going to see an increase in the variability in climate. We can already see, aspects of like extreme heat waves and extreme cold snaps and things like that. And those things are probably going to be super important for understanding disease dynamics, because I guess as you've seen in the research that I presented, these extreme climate conditions are the ones that are the most stressful to the hosts, and also have probably the most severe impact on the physiology of pathogens. So the better we can do at predicting these extremes and like the sort of pattern of occurrence of these extreme climate conditions, I think the better we're going to be able to do at predicting when and where we might have disease outbreaks that are triggered by climate events.
Thanks, Corey. Brian, what are your thoughts?
I agree 100% with Corey, and I think that variability is going to be one of the most interesting challenges. For vector-borne diseases, they tend to have a distinct seasonality, and this is often driven by the relationships between vectors and climate. And so Lyme disease, for example, is most prevalent midsummer due to the activity of the nymphal life stage of the black-legged tick, which is the main vector to humans. And we're seeing disruptions to that sort of consistent pattern of seasonality as a result of warmer days in the spring, causing ticks to emerge earlier. And this is making some of these vector-borne diseases less predictable, more difficult to educate the public about risks because of changes in timing, even more difficult for physicians to make accurate diagnoses based on historical relationships between vectors and climate. And so I think that variability is going to be one of the big challenges. Also, I think, as Corey indicated, some of the hardest things to predict may be the most important. And another phenomenon we're seeing with vector-borne diseases is that there are many that are changing in distribution, often as the result of a tick or a mosquito getting introduced to a new area for which it wasn't realized that that was a suitable habitat, a suitable climate, and then becoming established there and becoming an important vector tick-borne disease in a new region. And they may become even better established as climate changes in ways that may be more favorable for those invasions. And so those are challenging to predict, but as they're happening around us, we're starting to realize how important they are and focusing efforts on that.
Thank you. So another general question. Is climate change a major factor driving emergence of infectious diseases? That kind of follows on what you were saying, Brian, now or in the future. And let's hear from Brian first and then go over to Corey.
Yeah, it's a difficult question to answer because there are likely many interacting factors that are simultaneously contributing to the emergence of vector-borne diseases in general. And so globalization, landscape change, climate change, all these things occurring simultaneously has the potential to contribute to the emergence of infectious disease. I'm certainly under the impression that in a number of wildlife systems, it may really be playing a major role in expanding distributions of pathogens associated with wildlife. For humans, a lot of the work to date, I think, is based on modeling potential relationships between climate change and diseases that we expect to be particularly impacted by changes in climate, like mosquito-borne diseases such as malaria. And there is some evidence that climate change is contributing to these emergence of disease already.
Yeah,
Thank you, Brian. And Corey, what are your thoughts?
yeah, I would totally agree with what Brian said so far. And I guess like echoing what he said about changes in distributions, also changes in phenology of hosts and pathogens that could potentially put novel combinations of hosts and pathogens together for the first time. Phenology being the sort of annual cycle of things like breeding and flowering and annual events that occur in wildlife. Those could, so put together new hosts and pathogens in combinations that sort of are able to spawn new invasions of diseases in ways that I think are also going to be difficult to predict ahead of time. And I guess one other thing I was thinking is that, the root cause of global climate change is, human activities that are causing increases in emissions. And those are the same sort of root causes to other sort of global change scenarios that we know are causing the increase in emerging infectious diseases like globalization, and human movement. So I think they all have the same root cause, whether or not there's super strong relationships empirically yet with climate change. I think we're still working on that, but I'm going to guess there's going to be lots of links there.
Okay, thank you, Corey. And there's also a question that came in from the University of Florida, and it's really for you, Corey. Could you talk about, could good fire, quote unquote, good fire management be an effective tool for achieving some of the goals that would reduce BD or BSAL? Kind of like a merging or nexus of the two presentations.
Yeah, I think that is a really interesting and great question, and one that I hadn't considered. But given that one of the outcomes of our model suggests that increasing the number of thermal refugia, or the availability of thermal refugia for amphibian hosts, a thermal refuge being a place that they can go and sort of warm up, which usually allows the immune system to be activated a little bit, and also is generally worse for the BD pathogen because it prefers cooler climates. That suggests that perhaps, fire regimes that would allow for warmer microhabitats outside of the ponds could actually be one way of reducing transmission, just by helping amphibians outside of the pond clear those infections. So I think it could be that there is sort of a nexus between the two topics, which is interesting to consider.
Thanks for that. So another general question for both Corey and Brian. What can the DoD do to eradicate the pathogens under study or prevent their spread? So let's hear from Corey first, and then from Brian.
Yeah, unfortunately with BD, it's going to be really hard to eradicate this pathogen because it seems to be everywhere, everywhere we've looked. So we have to focus now on controlling its spread and perhaps controlling its transmission, especially to highly vulnerable host species. And that's some of the things that I talked about as, management implications that our models are pointing to. For the BSAL pathogen, which is not here, obviously making sure that it doesn't get here is going to be a really important thing, because I don't know how many of our listeners are aware, but the United States is the global hotspot for salamander diversity, and that BSAL pathogen has been more pathogenic and caused more declines in salamanders than in frogs, which the BD pathogen is actually the opposite. So making sure that BSAL pathogen doesn't get here should be a high priority. And doing that really means not moving anything that could be infectious around, and especially not from places like Europe where the BSAL pathogen is known to be. So not moving wildlife, not moving aquatic samples or things that have been in those ecosystems. If you've been a researcher working in an ecosystem where that pathogen is, obviously the Batrachochytrium pathogens can be killed by drying completely, but they can be, we think, moved around by things like wet boots or wet equipment. So making sure that things are sterilized before they're moved from place to place in order to kill that pathogen is a really important thing. And there's a whole group of amphibian researchers that are working on strategies to prevent the invasion of that B-cell pathogen into the United States.
Thank you. And Brian, your thoughts?
Similarly, preventing spread of ticks and tick-borne pathogens is difficult because they're strongly associated with wildlife hosts, and their movements into new areas are largely driven by independent movements by wildlife hosts. There are certainly some aspects of environmental management that can benefit reducing tick abundance, and there are even interventions in scenarios where tick-borne disease is particularly problematic, such as environmental use of pesticides, as well as applying topical pesticides to wildlife, which is a really interesting strategy that involves things like bait stations, where, for example, white-tailed deer come in to feed on a bait and are delivered a topical pesticide in the process. And that can be used for some effective local control of ticks. But that's very laborious and both time and resource expensive. And so in many ways, what we're looking for are more large scale activities that may integrate well with other environmental management goals that allow us to control ticks at the same time. I do think land managers will want to be on the lookout for our new invasive tick, the Asian longhorn tick, which is spreading through the eastern US and I believe has been reported in 17 or so states now. It's strongly associated with domestic livestock, and so installations that allow grazing by cattle may particularly want to be aware of its presence. But it can parasitize wildlife hosts as well, and so much of its spread is likely driven by independent wildlife movements rather than transport of domestic animals. So it's a shifting landscape, and staying abreast of what are the new threats, I think, is an important part of trying to stay out ahead of some of these emerging infections or vectors, since there's new ones popping up.
Thank you. One last general question for both of you. Why is more research effort needed at the nexus of climate change and infectious disease? Let's hear from Brian and then Corey.
Yeah, I think the short answer is we've really just started to understand the complexity of these relationships, that there's more than just direct effects of climate change on infectious disease emergence. And so being able to understand the indirect pathways by which climate can interact with disease risk will require more attention. We see evidence that there's many potential both biotic and abiotic factors that may be changing along with the climate that could then alter infectious disease risk.
Thank you, Brian. Corey?
Yeah, I would echo many of the same things. Many of us who study infectious disease have been taught about this disease triangle that includes the host, the pathogen, and the environment. And it's challenging enough to figure out how those three things, interweave and interact with one another when each of them are, stationary or stable. But when you start to get environments changing, you start to see impacts on host and pathogen changing as well. It becomes an ever-moving target. And so the more we can understand how climate change is going to work and how it's going to impact either one host or pathogen, the more we can start to put together the full picture of what diseases are going to look like in this shifting environment. I think it's a really challenging thing that, as Brian said, we're just starting to get a handle on how to even tackle these complex questions.
Well, thank you so much, Corey, and thank you again to both of the presenters we had today, going over their research and their results. We very much appreciate their time and efforts that they put into putting together their presentations. Our next webinar will take place on Thursday, July 8th in the Weapons System and Platforms Program area. 3 speakers will discuss energetic materials and munitions, including Dr. Jay Porritt and Dr. Anthony Shaw from the U.S. Army and Dr. Andrew Innen from the U.S. Navy. Registration is open, so please visit the SERDP and ESTCP webinar webpage to register for this and other future webinars. Before we conclude, I would like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you would like to refer to them in the future. We would appreciate it if you could take a moment and complete the survey that will pop up on your screen at this time. This concludes today's webcast, and thank you to everyone.